in our earlier lecture we have shown that each and every parts of our body faces that mechanotransduction by different mechanical forces. In this lecture, we shall go through that how that mechanotransduction happening in case of cancer and in case of bone tissue forming cells. Why cancer? Because in the cancer metastasis, mechanical force playing the main role for the migration of cancer cell from that source tumor to the distant places through that blood circulation system. And bone is a mechanical sensitive system, and it is bone forming ability is solely depends upon that mechanosignals. Without these mechanosignals, bone forming cells will not work. So, that is why we are concentrating on that cancer metastasis and bone forming and osteoblast mechanotransduction in this lecture. So, take home lesson of this lecture: utility of microfluidic platform for studying mechanobiology, particularly in cancer milieu, because we know that focal adhesion and lipid rafts and stretch activated ion channels are the mechanosensitive cues or mechanosensitive parts of the cells, and these are responsible for various biological outcomes of the different types of cells. Here we are focusing that how that focal adhesion complex dissociation and lipid wrapped microdomain internalization mammalian cells happening in the microfluidic confinements, and how that lipid wrapped microdomain organization in stress adaptive mechanotransduction in circulating tumor cells. Basically, what I want to mean that circulating tumor cells, when the tumor cells is in the bloodstream facing that shear stress of that blood. And as already I have mentioned that bone is the mechanosensitive organ, and how that shear stress influences osteoblast cells, means your bone forming cell, to work in terms of that calcium signaling. Now, we are coming that tumor formation, but how that cancer cells situation in the tumor. So, cancer cell starts due to the genetic aberration, and due to this lot of signaling pathways are starting, and one of the pathways are epidermal growth factor, RAS and ERK pathway. So, this pathway stimulates the proliferation of cancer cell, because we know that cancer cells have a uncontrolled growth rate. In that sequence, this helps that cytoskeleton arrangement, and it develops cytoskeleton pressure. So, which stimulates that Rho, ROCK and myosin light chain phosphorylation, then actomyosin interactions. So, these two pathways interact with each other to stimulate that extracellular matrix stiffness. As extracellular matrix stiffness increases, that stimulates that focal adhesion, which acts as a molecular clutch. It influences that intriguin activation, focal adhesion kinase, then stimulate both proliferation as well as cytoskeleton tensions. So, what will happening in that process, that ECMs get stiffer, means rigid. In the down here we can see that in the normal cases that ECM is soft matrix, cells have a normal size, when cancer cells start growth, uncontrolled growth, that matrix becomes rigid, and at the same time lot of other events happening in that situation, because as cancer cell grows, it invites that fibroblasts, that lot of immune cell like T cell, B cells in that microenvironment. So, cell population is very much high within this confinement. So, this you can compare like say one argon balloon is attached to a thread, and when that argon balloon is expanding, that tension in the thread is increasing. So, this type of mechanism is happening in that cancer tumor case, and at the same time that your blood capillaries becomes necrotic, means irregular, your extracellular matrix stiffness are not regular, means different places spatiotemporally ECM matrix density is different places is different. So, this is that multi-cascades of how that cancer cell metastasize or migrated from that source place to the distant place. At the source place means where that tumor develops, that metastatic cascades cancer cells within a primary tumor acquire that invasive phenotype. What invasive phenotype means? That initial positions cancer cells are anchorage dependent, means they are more or less static. During the high population of the cancer cell and other cells, and due to oxygen limitation and so many other factors, that cancer cells, some cancer cells want to escape from that site. So, for that escape mechanism, they have to develop some non-anchorage dependent system by which it will be changing to metastatic cells, means from that primary site they will be invading that nearby blood vessel or lymphatic vessels. So, as it enters the blood vessel, it acquires the property of non-anchorage dependent property, means they need not any anchorage for their survival, they will survive in the blood flow and faces that shear stress of the blood. So, in that shear stress, majority of the your tumor cells will die, some will survive which can withstand this shear strain. Then in that way migrated to a distant place, that is a distant organ CTC exists in the circulation and invade that microenvironment of the foreign tissue, means the distant tissue, and there cancer cell must be able to evade the innate immune system and also survive as a single cell or as a cluster of cells. Then again that tumor starts. Basically, starting from that source tumor to blood vessels, this is totally driven by that mechanical force. So, mechanotransduction is playing very vital role in that migration process, and interstitial flow is playing major role when that metastatic cell migrates from that source and invade that near about or near blood vessel. So, interstitial flow depends upon that pressure gradient develops between arterial interstitial space, that near about lymphatic vessel and that venules. And as that tumor is very much heterogenic with respect to ECM, say interstitial pressure difference is also different in different places. So, microfluidic platforms are used extensively to study the migration of the cancer cell in that interstitial flow system. So, what literature is saying that in the interstitial flow affects cell migration in different ways: local strain gradient induction, flow of regulated proteolysis by migrating cells, stress mediated cytokine activation, autologous chemotaxis. Main findings are that downstream migration means that in the direction of the interstitial flow, that is your cells are migrating according to the chemotaxis, means your growth factor or cytokine gradient, whereas lot of cells also migrated upstream of that interstitial flow, and that is driven by stress activated things, that is focal adhesion is involved for your upstream movement, means both type of movements are available in that is your from tumor primary site to migration to nearest blood vessel. And as that tumor is heterogenic population of cells and cancer cells itself are heterogeneous, so obviously their migration will be heterogeneous. To understand that system, Hessler has utilized a new platform for microfluidic system, where that two types of interstitial gradients are exploited to know the heterogeneity of that cancer cell migration. So, heterogeneity means some may respond to flow by moving faster, but randomly. Some cells migrate in the flow direction into autologous chemotaxis, and others may move in the opposite direction due to the local stress gradients, which means there are different types of cellular migration in interstitial flow. So, what Hessler finds that cell migration within a population is heterogeneous, that is some cells migrate upstream and other downstream of the flow, and minority of the cell populations are more random. There may be more directed, more randomness, more faster, means these are the around 5 to 10% of the cancer cell populations are very much heterogeneous with respect of their migration rate. So, these cells might be the important for the metastatic survival or metastatic potential. So, with this study they have identified that migrating potential or different cells with respect to other majority cells which have a different migration potential. So, with this background, our collaborative work with Professor Suman Chakraborty has revealed some unique findings when that CTC means your at that encourage independent stage cancer cells migrating from that source to that bloodstream, like say focal adhesion and that lipid raft, how they are behaving and related processes. So, challenge in that system is that it is to be addressed whether environment of surrounding the micro confinement plays a vital role in the cell survival within the passage of vasculature or tissue matrices. So, how will we address this problem? In our earlier lectures, we have described that how to measure the traction force by using traction force microscopy system and membrane fluidity and other microfluidic platforms to reveal that augmented stress responsive traits of cell lines in a micro confinement. So, in that process we found a new parameter, like say we are telling that cell response time TR is induced to analyze the stress adaptive properties of the mammalian cells. What is TR? So, it is the time lag between focal adhesion disassembly and lipid raft appearance in the same positions of the cell. So, now we are going to details how that phenomenon is happening. So, here is the image of one mammalian cell, then this is the image of lipid raft in the cellular membrane as well in the cellular inside of that. and these are the images of that focal adhesion points, and we concentrated on one of the focal adhesion points, that how it is dissociated. When you are giving shear stress, that focal adhesion points in that where that shear stress is given, that focal adhesion points dissociates. You see that here this focal adhesion slowly dissociates, and that dissociation, for the full dissociation it takes around 20 minutes. It is represented by this curve. At the same time, we are looking for that how lipid rafts behavior in that situation. Here we see that in the same confocal plane, that lipid rafts internalization, it takes around say 20 minutes or 18 minutes time. So, this is that time lag between the two events, and this is a characteristics property of that mammalian cells when you are giving some stress, means in the condition of fluid flow. So, now you want to know, so how that focal adhesion is involved in this process, and this is that representation where you are showing that 50% of the lipid raft internalization events happening at any given period of time under that some shear stress. When we inhibit the focal adhesion by using focal adhesion kinase inhibitor NSC 23766, we are finding that red pillars basically they are indicating that lipid raft internalization event are not happening. So, we can correlate that focal adhesion dissociation and that lipid raft internalization. This is a characteristics phenomenon in that confinement and in this shear stress, and that response time TR dependent upon that two variables. One is your confinement. What I want to mean here, that height of the channel is to height of the cell. Height of the cell is constant, we can vary the height of the channel. So, confinement one parameter, another is shear stress developed by the different rates of fluid flow. So, there is a critical conditions like say when that channel height is 70 micrometer and that shear stress is more than 10 dynes per square centimeter, then we are finding that sigmoidal decline of that TR, but in other cases this is not behaving, or open system this is not behaving sigmoidal. So, we are taking these critical conditions and we measured that TR for that varieties of cells starting from that normal cell lines like say NIH33, L929 and most evasive cancer cell line like say MCF7, B16F, HeLa cell and lot of other cells. And if you see that what we are seeing in that micro confinement situation, at that shear stress 10 dynes per square centimeter and TR is around 10 minutes for these cancer cells, and whereas it is more than 20 to 30 minutes for the normal cells. And that indicates that we have find a unique parameter response time to differentiate between that normal cells and cancer cells, particularly in that micro confinement where that is critical micro confinement and that critical shear stress. So, now question arises how this factor is happening in the molecular level. So, we know that epidermal growth factor is a growth promoting one of that growth factors, and it binds with that epidermal growth factor receptors and pass the signal, means growth factor receptor is phosphorylated, then signal is passing. And the lipid raft is very much connected with these membrane bound receptors, and on shear that lipid rafts get rearranged and undergoes internalization. So, what we find it here that autocrine growth factors, that EGF works nicely in that confinement with this Western blot and that imaging techniques. We saw that in absence of EGF, only shear stress is suffice to activate that EGFR, and which helps the internalization of the lipid raft, and at the same time that is your focal adhesion dissociation. So, we connect these things, that in the micro confinement autocrine effect of that EGF stimulates that chemical signal transductions, which is influenced by that mechanical signals of shear stress. So, what we infer from these studies, sigmoidal sepal decline of TR depends on confinement and shear stress. That same TR occurs when the channel height decreased below threshold value of 70 micrometer and stress is elevated beyond 10 dynes per square centimeter. And this TR is due to augmentation of secreted autocrine growth factor, epidermal growth factor, that is EGF concentration, and amplification of the fluid shear stress, the microfluidic environment. And TR is less for invasive cancer cells, means cancer cells are stress adaptive. They can adopt, which cells adopt this shear during their blood flow, they can survive, otherwise they will die. So, these things indicating that cancer cells are more adaptive to stressful situation in the micro confinement of blood vessel by arranging their cytoskeleton during migration and extravasation. With this context, we are following that another set of experiments where involvement of lipid raft in mechanotransduction is explored in details, because lipid raft is that place where it can gives the shelter of your membrane bound enzymes, growth factor receptors, stretch activated ion channels, and it also activates the downstream of mechanosignal induction, that is MMP pathways. So, here we are exploring that autophagy, another biological biological readouts, or it is a phenomenon of housekeeping mechanism whereby the cell degrades damaged unnecessarily dysfunctional cellular components through the action of lysosome. What is lysosome? Already we have discussed. This is a your garbage bag. It recirculate or digest that unwanted material and do its job. So, autophagy is a phenomenon where that unutilized organelles or molecules, so and so forth, they are digested in the lysosomes and gives the energy to the cells when the cells are in stress. And different types of autophagies are there, like say mitoautophagy, nucleoautophagy, macroautophagy, etc. nowadays it is coming. So, autophagy is a double edged sword, means when that cells are in stress condition, like say when that metastatic cells are feeling the stress in the bloodstream or cells are in starvation, they utilize its own energy sources and metabolize it or digest it, then metabolize it to get the energy. But that is your pro survival mechanism. On the contrary, when that autophagies are uncontrolled, means self eating things going unrestricted, then it kills itself, it triggers that apoptosis, that is programmed cell death. That is why that autophagy is a double edged sword. It acts as a pro survival or death inducing mechanism according to the need of the cell. So, now we are looking for that how that mechanotransductions or shear is involved to stimulate that autophagy, either pro survival or death inducing. So, that is why you have raised the questions whether physiological shear stress elicit autophagy in cervical cancer cells as immediate adaptive response, whether this response is protective towards the cells, or do membrane cytoskeleton components like say lipid rafts participate in the process of shear mediated mechanotransduction. So, in that experiments we have two parts, means on chip part, another is off chip part. On chip part we are giving that cells with shear stress, and with that measuring the autophagy by reporter plasmid PTFL LC3 exposed to pulses of steady flow, and off chip part we are using that syringe pass model for the application of shear stress to cells and collection of cells, then after that assess the viability and protein expression and etc. So, what we are finding it here? So, in the control set means when we are giving no shear, we are measuring that autophagy in terms of puncta. Puncta means you can see the dots actually, say green dots and yellow dots and red dots. So, the autophagic flux is the ratio of that red and green dots with respect of your total number of dots present in that in one cell, because we are measuring that autophagic flux of single cells taking say 100 cell or 1000 cell together, and we are averaging the data. So, this data reveals that when you are giving 2 Pascal shear, that yellow dots means is a mixture of your green and red dots, and only red dots are increasing with respect of your total dots with green dots. And when you are giving MBCD, which disrupt that your lipid rafts, that with this 2 Pascal shear, surprisingly we are seeing that there is decrease of significant decrease of your RFP and GFP means yellow dots. So, this indicates that MBCD which disrupt that lipid rafts definitely involved in autophagy flux. So, next question we are arising, how that shear stress induces the autophagy flux, whether it is a pro death or pro survival. Here what we find in a nutshell from that slide, when you are giving shear, that autophagy is survival inducing, means that it induces cell to survive in that stress situation, means stress adaptive. And when you are disrupting that lipid rafts, it is inducing more apoptosis, more cell death, means that with these findings that lipid rafts has a interesting role to withstand the shear and gives the particular biological outcome with respect to autophagy, whether it is a pro survival or death inducing things. So, all these things is modeled in that way, inhibition of protective autophagy by lipid raft disruptions and inhibition of P38 MAP kinase phosphorylation is the intermediate of that signaling chemical signal processing, short of lifespan of tumor cells in circulation and lack the metastatic ability. Next we are coming that bone system, it is a mechanosensitive system where that mechanical signal is necessary for that activity of bone cell osteoblast. So, here we are saying that this is the blood vessels surrounding cells are osteoblast cells, then that bone formation is going on when then the osteoblast is conforming or converted to osteocytes. During this whole the process, that is mechanical stimuli influences lot, means when normal state mechanical stimuli is there is a equilibrium between bone dissociation and bone formation, that is the used state. When overused means when you are using working or more load in the bones, indicated by these arrows, then osteoblast recruited and it induces bone growth, then it is coming to a steady state. Then when it is underused, means not much mechanical signal is going on, then osteoclast bone degrading cells are activating, then bone start degradation, then is some steady state reaches. So, this indicates that mechanical signal is very much responsible for osteoblast function, that is bone forming function, and it produces a lot of your extracellular matrix, that is your calcium related calcium hydroxyapatite and etc. So, what literature is saying that when you are giving shear stress on osteoblast in parallel channel on chip, intensity of the intracellular calcium intensity peak was proportional to intensity of the shear stress between 0.03 Pascal to 0.24 Pascal. And this shear intracellular calcium concentration shows peak at the beginning and influence the mechanical transduction. And when you remove the shear, then again calcium concentration decreases, and that calcium intensity peak, it depends upon the shear stress. So, it was reported by that cow. Then after that, our group with collaboration with Professor Chakraborty, we are utilizing that fluidic platform to explore that how the different types of fluid flow influence that your osteoblast function in terms of calcium flicker generation. So, here in the fluidic channel, we are using different types of fluid flow, like say steady state, pulsatile flow and oscillatory flow in the osteoblast cells. What we are finding that oscillatory fluid flow, that is oscillatory fluid flow shows that maximum calcium flicker concentration as well as calcium amplitude, and this is measured by your calcium cytoplasmic calcium flicker concentration, and calcium flicker is due to that your calcium transporter channels which are present in the membrane, plasma membrane, endoplasmic reticulum. So, now question arises, what type of that is stress responsive channels or what type of calcium channels are involved in that situation of shear stress. So, you are using so here we are seeing that your oscillatory shear stress is involved in the process, and what frequency here that 0.2 Hertz frequency, that oscillatory shear stress is the maximum showing that calcium flicker concentration in that the osteoblast cells. So, to explore that what type of calcium channels are involved in the shear situation, because varieties of calcium channels are involved in that process. So, we use different types of inhibitors for particular group of your calcium activated channels, like say voltage gated calcium channel, store operated calcium channel, stage activated calcium channel, calcium ATPase and phospholipase. And there are specific inhibitors for these type of channels, and what our findings is that when you are using gadolinium chloride, which is inhibiting the stress activated calcium channels, that calcium flicker concentration decreases drastically around say 80%. So, we are telling that stress activated calcium channels are involved in this process, and we also know that calcium in that stress activated calcium channels are harboring in that lipid raft. So, definitely lipid raft is responsible or activated or organized in that when you are giving that oscillatory shear stress. So, to identify that which type of stretch activated calcium channels are responsible. So, these group of calcium channels are termed as transient receptor potential superfamily member. So, there are different members like say V2, P2, M7, M6 etc. So, what we did, we knocked down the osteoblast cells by siRNA technology, then we are looking for that, or we are confirming that whether cells are knocked down or not by Western blot, and we are saying that TRPM7 all these things which are successful knockdown for the cells. And in the knockdown cells we measure the calcium flask in that oscillated shear stress, and we find that the TRPM7 knockdown cells there is no calcium flicker. This clearly indicates that TRPM7 is really responsible for that calcium flicker generation due to oscillated shear stress. So, as we mentioned that lipid raft is the position, or lipid raft is that means domain where that the stage type of stage activator ion channels are present. So, we are looking for that how that spatiotemporally in the membrane that lipid rafts and that calcium flicker generation is occurring. So, what we are seeing it here at that same time point, that same time point and that same places calcium flicker generation as well as lipid raft organization is happening. So, obviously question arises, so what is the role of your lipid raft for calcium flicker generation? So, if we disrupt that lipid raft by using your methyl cyclodextrin, what will happen? So, if our hypothesis is correct, then if we disrupt that calcium flicker generation will not be there. So, that is demonstrated here. So, if we use MBCD and do that same shear, and calcium flicker generation is almost the control level. So, this indicates that shear oscillatory fluid flow induces that lipid raft rearrangement, which induces TRPM7 organization, and when the TRPM7s are come closer together, that induces calcium flicker generation inside that cytoplasm. So, all these things are possible using that microfluidics to understand that mechanoid signal transduction during cancer metastasis and osteoblast cell function. So, what we are exploited here, high resolution imaging at single cell level to study that spatiotemporal dynamics of intricate membrane related cellular events. So, what we find is here that disassembly of focal adhesion points due to shear triggers localized lipid raft internalization, and shear stress triggers calcium flickers in shear exposed bone cells. Simulation of physical flow conditions mimicking in vivo niche physiological laminar shear stress induces EGFR activation making cancer cell invasive, induces protective autophagy adding cancer cell survival, and oscillatory shear stress of frequency 0.2 hertz induces optimal bone growth, means your calcium flicker generation it will go. And mimicking in vivo confinements possibly replicate the natural cells confinement aspect ratio, and our finding is that cancer cells are more active, have lesser response time, means they are more adaptive than non-cancerous cells in that confinement. So, such studies hint that importance of lipid raft mediated mechanobiological signaling cascades as a candidate drug targets in various physiological ailments.